All right, thank you. Oh, I am on. Thank you all for coming. A few housekeeping items. This is being recorded in the back, so you're all aware of that. If you're interested in getting slides, they are available on the ICTR website. So you can log into that. If you set up an account, you will get emailed on future things that are put onto that website. In terms of the internet access here, you should be using the Horton Grand, and the login is, or the password is 311 ISD, 311 ISD, which should give you access to the web. If you need to go to the bathroom, those are right out, so basically go that way, which involves a quick jog to the left and then a quick turn to the right, but they're basically right behind that wall in a little hallway. Let's see, attendance is enough that I can kind of know. How many of you are clinicians? Clinicians, 1, 2. Statisticians? A couple of those. Okay, so a mix, that's all good. I just wanted to kind of get an idea what we were talking to. What we're going to do today, we're going to go through a number of topics. Some of the structure is going to look like the webinar that's been posted online, but we're going to add a lot more meat to it as we go forward. So a quick description, this is also part of the webinar that was online, kind of talking about what a gold standard RCT is. So keep in mind, typically this is a two-arm trial, treatment versus control, has basic elements of randomization, and all of this is designed and has been done for about 60, 70 years or so. When we're talking about innovative trials, by necessity, we're talking about breaking a few of those components or doing them differently. And so what we're going to do today is we're going to talk about a number of innovations, going to talk about the motivation for each one. As opposed to the first webinar, we're going to give examples and some main ideas of each one of them. So we're going to start to quantify some of what they do and why. And certainly all of the trade-offs involved in this. So if you pick an innovation, sometimes there's, quote, a free lunch. Other times you have to make sure that, account for the sample size might be increased, the power might be decreased, and you have to weigh the trade-offs involved in that. So keep in mind, modern RCTs go back to the Streptomycin trial in 1946. There's a publication that came out in 1948 describing that trial. Really kind of the last innovation is randomization. Prior to this, people either used alternating assignment or pretty much assigned things at will. And at some level, this has been rather, it's been stuck at this point. This 1948 trial is run over and over again, and in lots of ways we have advanced. And so we want to talk about the ways that we've advanced. Certainly a standard RCT does well. So we want to make sure that deviations are for a reason. So again, we're going to talk about what are the features in this gold standard, what are the negative features, what are the trade-offs. The main components here, we have a lot of components that are designed to avoid bias. These include randomization. Randomization is kind of a catch-all. It avoids biases on essentially anything you don't know. We also routinely stratify. That's intended to directly equalize groups on biases that we may know in advance. We also employ blinding, and often we have a fixed sample size, so we avoid the investigator being able to kind of look at the data until they get the answer they want. So all of these things avoid biases. The rest of this is aimed at obtaining interpretable results. So we want to make sure we often do a two-arm trial. That's the sense that if we see a difference between groups, we'll directly attribute it to the two groups. We randomized, we saw a difference. The only thing they differed on was the two arms. This caused the difference. We often try to look in a homogeneous population, again, trying to avoid the uncertainty. We use lots of validated standard clinical endpoints. All of this is really intended to make things as simple as possible in the trial so that we can get clean answers. The trouble is that they're very expensive answers. So we often have to do hundreds population. That's effectively all you can do. It also asks us to look at really narrowly focused questions. So if I say you only have 2 arms, what's the population? Oh, I'm going to focus on this group of people, when perhaps, I really want to know, well, maybe it works in everybody, but maybe it only works in this subset. And so these are conducted you have 1 or more interim analyses. I'll give an example in a moment. And at each of these interims, we can stop, make a decision, we might continue, look at the data again, we might continue, so on and so forth. As I said, these have several titles: futility stopping, group sequential, sample size re-estimation. A Goldilocks design is an alternative here, which is intended to account for incomplete information at each of the interims. All right, so an example of this. Suppose that I'm testing a dichotomous response, so everybody is yes or no. And suppose that my control, I anticipate a 30% response rate. And in my treatment, I'm expecting 50%. This is my hope. So going in, I'm powering the trial for a 30 versus 50% effect. A standard trial here might have about 100 patients per arm, gets us 83% power. So a standard way of conducting this is I would enroll 200 patients, 100 in each arm. I would do at the end of it a standard kind of normal test. If my p-value is less than 0.025 one-sided, then I'd say, hey, the treatment is efficacious. Okay, well, that requires 200 subjects. I'm going to try to convince you in a moment that what you've just bought is a horribly expensive insurance policy against bad luck, and you typically don't need it. So let me ask a question. I'm going to take a poll here. What if we actually observe? So this isn't what if the truth of the drug. This is what if I actually get in my trial 30 out of 100 responders on control, and I get 50 out of 100 on treatment. So I powered it for that effect. Suppose it actually happens. Suppose I actually get that effect. What's the p-value? More, equal, or less than 0.025. So I powered it to get significance at this level. How many people think it's more? How many people think it's equal? How many people think it's less? How many refuse to answer? Fair enough. All right, it's actually really small. It's about 0.0016. And that's for designing a trial at an 80% power. If you design a trial at 90% power and you get the observed effect, the p-value is about 0.0006. So if you actually get what you expect might happen, you're far into the realm of significance. So why did we need 200? What were we really doing this for? I had an interesting conversation, CEO of a small biotech. We were doing a kind of adaptive design trial, and he asked me the question, why did you pick 200? Happened to be the size of his trial as well, not the same as this. But in any case, he was an engineer. For what it's worth, I broke 5 generations of engineers to become a statistician, much to the disappointment of my father. So anyway, I have these conversations a lot. Essentially, and interesting, I'm putting some words in his mouth, but I think what was essentially the expectation here is that I picked 200 because somehow I knew when the p-value would be significant. That somehow I was going to get a pattern like this. So at n equal 100, the p-value is 0.27, and as I go to 150 and 200, 200 is kind of the magical time where the p-value gets under 0.025, and I picked 200 because I knew that's what would happen. And you certainly can get data like this. But in general, data can come in lots of forms. I didn't cherry pick this particular example. I just basically sat on my computer and flipped coins. And this is also a pretty standard trial that comes up. P-value is 0.0015 at 100. I'm already significant. And it's tiny at 0.000001 when I get to 200. So I'm in a position, I didn't need all those patients. I'm incredibly significant. And certainly what I did is I flipped these coins under the assumption of 30-50. This is what I powered for. This is what all statistical methods said you need 200 subjects. Well, in this case, I didn't. I got it a lot earlier than that. Here's another one. This is one, it's again, I'm flipping 30 versus 50. This one never gets to significance. We got the 0.1, 0.06, 0.19, it's going up and down. This is a trial that we know that if it has 83% power, 17% of the time you don't get significance. This is one of those 17%. All right, so what happens in lots of these trials? I just gave you 3 and showed you what happened at 100 to the p-value, at 150 to the p-value, and at 200 to the p-value. So I generated 50 trials. And all of these lines, I have plotted each of these lines. This is the p-value over time at 100, 120, 140, 160, 180, and 200. All of these are simulated 30% control, 50% in treatment. Now, one thing that's certainly going on is all of these lines are going up, not uniformly, so some of them go up and down, but the general trend is up. The other thing that's going on here is these are the lines of significance. That's for 0.025, 0.01, and 0.001. I'm plotting the z-score on this axis, so I didn't put lots of tiny p-values in my graph. So what happens here? Well, at 100, you can see a lot of these are already significant. So, and they become highly significant. At 150, even more are significant. So most of these, you can see there's a whole bunch of trials. They're significant, and they're significant far before I got to 200. So why did I get 200? What I've done is I've bought an insurance policy. I've picked 200 not because that's when the p-value is going to get above 0.025. What I'm waiting for is the slow ones. So like this red one, I'm waiting for trials like that to finally have gotten above that line of significance. And I've chosen that 200. When I say I have 83% power, 83% of the lines are in the significance region above 0.025, and another 17% are below. And what I'm doing when I choose a sample size of 200 is I'm waiting for that to occur. I'm waiting long enough for all of these laggards, and I'm not taking advantage of the fact that most of the trials have been significant for a long time before I got to look. All right, so a group sequential is a way of handling this. We have to pick interim analyses, and you can pick a couple, 100, 150, 200. You can pick many, 100, 120, 140, 160, 180, 200. So I can pick lots of interim analyses. Generally, the more you pick, the more efficiency you get. there is diminishing returns. So one thing we often do is we basically calculate if you do 2, if you do 3, if you do 4, if you do 8, here are the results. And you need to decide how complicated it is to run an interim versus the statistical efficiency involved. We've had trials where it's incredibly easy to run an interim and you may as well run tons of them. Other times, we got to have a huge DSMB meeting, there's all kinds of reviews, and that may be an incentive to run less. So here is the kind of example that you might see, and this is 120, 140 up to, 100, 120 up, 140 up to 200. One thing that's going on here, you'll see at 100, I'm going to declare success if the p-value is less than 0.0031. I need a highly significant result in order to get that. Now keep in mind, there are a few trials that hit this. That's 0.001, so some are going to make it. At 120, it gets a little bit lighter, at 140 a little bit, and these are going up. And finally, at the end, I need a p-value of 0.0183. The way I tend to think about this, if you go to like a carnival game, you're standing in front, they'd say, okay, you've got to throw a ball through a basket in order to win a prize. There's a certain size hoop that you got to throw it into. Ordinarily, if you're going to get one throw, I'm going to let you need a p-value of 0.025. There's a certain size hoop you have to get it into. If I'm going to give you multiple throws, so I'm going to let you throw it, how many is this? 100, 120, there are 6 different throws that you're going to get. I can't give you the same size hoop for every single one of them, because what happens is that increases your chance of getting one, even if you're a bad shot. So what all these are aimed at, the reason that all these p-values have changed, is because you don't get one throw at a big hoop, you're getting 6 throws at smaller hoops. It's designed so you don't lose any power, you don't lose any type 1 error, but it's designed to allow you to win this trial earlier. And again, the notion going back to this graph, what I'm doing is I'm just figuring out when this line finally gets high enough, I'm going to go ahead and declare success. I'm not going to wait for 83% of the trials to get there. I'm going to stop this trial for success whenever a trial actually gets high enough. It turns out in this particular trial, 28% stop at 100. And that's again 30 versus 50. This is what I'm powering for. So this is not for a dramatic effect. These are just ones that got lucky early. 12% at 120, 11, 11, 10. 7.9% win at 200. And then here is 18% that lost at the end. So these are ones that went through the entire sequence and never actually made it through any of these hoops. If I look at this, my power is 81.8%. Now remember, I was at 83.3. So the first trade-off here is I've lowered power. By looking earlier and by throwing through 6 hoops, I've lost a little bit of power by doing this. What I've gained is my expected sample size is now 148. Some of these trials stop at 100, 120, 140, they stop early. A few of them go all the way to 200. So I'm basically saving 25% of my sample size, and it has cost me 1.5% power. So I have to decide about that trade-off. I can actually pay that trade-off in a different way. Suppose I say that I'm just going to increase my maximum sample size. So now I'm going to do interims from 100 up to 220. So this is a possibility I may need a larger trial. But what does this do? So again, I've split this all out. You can see here's 20% of my trials. They've had to go to 200. Oh, this one. 21.5 reached 220. So I've made 21% of my trials longer at 220. And in addition, a lot of them are smaller. My expected sample size is 156. So if I have to fund a lot of trials, if I'm doing this, how many of you run more than one trial in your life? How many people have run more than 5 in your life? How many have run more than 100 in your life? Okay, there is a maximum. Okay, just making sure. By the way, I know an Alzheimer's researcher who's run like 100 trials and never had a success, and you really ought to be hitting the type 1 error rate at this point, so that's a little unlucky. But in any case, this is, so we're saving sample size on average. If I have to do this repeatedly, I have to go up to 220 every once in a while, but I get to go down to 140, 160 a lot more. So that's why I get to save sample size in doing this repeatedly. It's also worthwhile to consider futility. So suppose we were 140 patients in, and what we observe is 15 out of 70, that's 21% on control, and 19 out of 70, that's 27% on treatment. Where am I right now? I got a p-value of 0.2147. Okay, that's not bad. Certainly not 0.025. One question to ask is this trial worth continuing? So certainly if you were on a standard DSMB, I'm on a lot of DSMBs, we would look at this. In the absence of a rule that says to stop, what would the discussion be? Well, this isn't going as well as we hoped, but there's no evidence it causes harm. The trend's in the right direction. We have no reason to stop this trial. We're going to go ahead and continue it. So one question to ask is, well, how likely is it that we're actually going to win this thing? We currently have a p-value of 0.21. We know that we need to get that to 0.018. And if you could imagine applying this to a non-group sequential design where you have to get the p-value to 0.025, it would look very close. But here we've got to get to 0.018, and we've got to do it by 220. Okay. In order to get a p-value of 0.018, that requires about a 15% observed effect. Your friendly statistician can back calculate that for you. All right, so we need a 15% effect. We've got about a 6% effect right now, and we're 140 patients in. So at 220, in the next 80 patients, what do we need? Well, we need a 32% effect on those next 80 patients, and right now we're running at 6. So something's got to give here. Either we've been unlucky to date, and our luck is going to have to change significantly, or this might be a trial that we're just not on pace to win. We often use predictive probabilities in order to calculate this. So I can ask the question, what is the likelihood that this is going to win? There are two pieces to the uncertainty in that. In the next 80 patients, there's sampling variability. If I told you if Mother Nature came out of the forest and said the true rate is 40%, in 80 patients you're going to get 40% plus or minus. So that's sampling variability. The other thing is Mother Nature does not come out of the forest and tell you it's 40%. So you don't really know what the rate is either. So there's uncertainty in the parameter. What predictive probabilities do, and I'm omitting the details here, but can certainly refer to papers, and I'll be happy to write blogs on this or whatever people would like to see. But a predictive probability incorporates both of those uncertainties, and you can calculate the predictive probability of success is 6.9% here. It's quite low, and I hope that actually matches the intuition. 6% on the first 140, need 32% on what's left. That's a big shift. It's not very likely to happen. All right, so I'm going to add futility to the example that I just had. And at each interim, I'm going to stop for futility if the probability of eventual success is less than 5%. Now that 6.9 I just did, I would have let that go with this rule at 5%. I would have continued on to the next interim. The value that you pick here, this is something, again, your friendly neighborhood statistician ought to be computing. If I do 5%, if I do 10%, if I do 15%, what are the consequences? The things that you're trying to manage here, you certainly want to stop poor treatments. If things aren't going well because your drug is not working, you would like to stop the trial. You also Remember that even in good scenarios, sometimes you get unlucky. That's 17% of trials that didn't win. I don't want to go out to 220 for those. If I'm going to lose, I'd rather lose at 140 than lose at 220. It's just easier on everybody. The other thing that I have to do here is I don't want to declare futility too often, because every once in a while there's a trial that looks bad early that comes back and wins. And if I declare futility too aggressively, what happens is I'm going to lower my power because I'm going to eliminate that possibility. So here what I've done, make clear, we have probability of eventual success is at 5%. I incorporated that rule. And so now what you can see here is these are the same p-values up to 220. This is the probability of winning at each of the interims. And then there's a probability of declaring futility. There's a small number of trials, they look really bad really early. And so those 3% stop. And then just a few stop kind of at each interim. I want to emphasize that most of the trials that are stopping for futility, if you had let them go, they would have lost. Where again, we're trying to find that 17% that failed and get rid of them earlier. I have made a few mistakes. So remember, the power was 85%. Futility has cost me 1.7% power by putting it in. 1.7% of trials I stopped for futility that would have gone on to success. However, that 83.3, I'm now exactly the same as the trial you started with n equal 200 that had no adaptation whatsoever. I've increased the sample size, I've put on the group sequential, I've put on futility, my overall power from all of those things combined is the same. Only 10% of my trials are reaching 220. So this little extra that I spent, I'm not doing it very often, and almost 80% I'm actually saving. So certainly I'm trying to get rid of this insurance policy that I bought. And the expected sample size is 150. So I gained a little bit in this scenario. This is when the drug works. Futility is not designed for when the drug works, or it's designed for when the drug doesn't work. So what I illustrated here was that I didn't cost you much power. This is what happens under the null. I actually can stop almost 60% of trials at 100. So if the drug does nothing, 30% versus 30%, I'm out early. I can get rid of 80% of the trials at 140 or before. So I've still got 83% power in the alternative. I'm stopping on the null. The expected sample size here is 123. They're all out really early. Now, if I were having to fund a lot of trials, what does this do to my population? I took a quick survey on NHLBI areas and kind of what the success rate is in trials. Imagine that you had to fund a bunch of trials and 30% of the drugs work and 70% don't. Remember, trials, it's hard to develop drugs and have everything work here. If you were in that boat, what would happen? We've obtained the same power. The expected sample size here, in the 30% of trials that work, we averaged 150 subjects. In the 70% that didn't, we averaged 123 subjects. Overall, for this population, I average 131 patients a trial. Some of them are trials that work, some of them are trials that fail. The futility works better than the success stopping, actually. And you can fund 52% more trials than you could otherwise by doing this kind of thing. So again, the flexible sample sizes, the expected sample size significantly reduced, some numbers on that, 25 to 40%. I haven't talked about this, but I said it was so uncertain when significance occurs. If you actually didn't know it was 30 versus 50, maybe it's 30 versus 70, maybe it's 30 versus 50, maybe it's 30 versus 40, that increases the uncertainty all the more. you want to have more interims to account for that. And finally, you do have to pay for this. You have to account for the multiple analyses, those p-values, they can't all be 0.025. You may have to increase the maximal sample size, and also keep in mind if you have safety requirements, stopping early might invalidate those. If I need 200 subjects to get an adequate safety database, I can't do this. If I need to make sure I have adequate numbers of people in different ethnic groups, for example, this may be difficult to do. You have to make sure that those kind of operational concerns are also handled in this. So all of those are part of this group sequential. All right, I'm going to switch gears. My colleague will.
You want to ask questions?
Questions? Yes, sir. Oh, can we give you the mic for the recording?
Adam Edisalo from EMIS. So what we find is DSMBs, as you noted, do they listen to the futility and efficacy rules that you put in? How binding are these, and what type of information do you put in the protocol to make sure that the interim analyses and futility have a lot more context around it, so when they get to a meeting and they say, well, should we stop or not, there's more context behind what went into the boundaries?
So the question was essentially what goes into a DSMB, what goes into the protocol, what goes into the charter, how is this actually implemented? I was thinking three things. I'm going to start going through them. I'm going to end up with two. You know, there are three kinds of people, those that can count and those that can't. So anyway, one thing, certainly in the protocol, it needs to be very explicit. Futility will be declared if the predictive probability is less than 5%, and here is the calculation, either in the protocol or in the SAP referenced from the protocol. Usually at the kickoff meeting to the DSMB, if not prior, you also would have a discussion with the DSMB. If you're the sponsor, you want to make your expectations clear here. You know, this is the rule for futility, and we want this to be followed so that everybody's clear on that. The other thing that you would show is example trials. Here is the kind of data. If you're at 140 and this is the data that you see, these are our criteria with stopping. And so that's a reason, effectively, you'd end up saying things, look, if we have less than a 6% effect at n equal 140, we want you to stop. And that's what this rule says. And at that point, if the DSMB, if there's uncertainty, you can have a long conversation about it and nobody's blinded yet because there isn't any data. So you want to be upfront about what the expectations are. I have heard horror stories of, and academic settings are a little different. Private sponsors are really picky about making sure the futility rules are followed. I know of cases where the DSMB went rogue and essentially said, we kind of want to see what's going on in this one subgroup. So they really ran the trial twice as long after the futility rule had been hit. They have not been hired as a DSMB member very often anymore. But anyway, you want to make sure that those expectations, really it's about, you don't want this to be embedded. This is part of the discussion and laying it all out in advance and making sure everybody is bought in before anybody has to be separated. Oh, all right.
All right. So the next topic we're going to talk about is single-arm trials. This kind of falls under the same banner as how do we minimize our resources when we're running a clinical trial. So one way, as Kurt described, is to have a flexible sample size. Another thing we sometimes do is run a trial where all of our participants receive the investigational therapy. So these are what we call single-arm trials. There's no placebo arm or no control arm in the trial. So when we run a single-arm trial, what that really means is we're assuming that we have some information external to the trial that we're going to bring into the trial. So we've essentially made some assumption about what patients might have looked like if we had a control arm. So we've made an assumption about what our average rate is, about what our response rate is, assuming that that's what the behavior is for patients that would have been assigned to a control arm. So this might come from historical data, for example. Maybe we've run another trial in this population. It's common to do this in rare diseases. One of the big reasons that we might consider a single-arm trial is that patients are hard to find. So if we have a hard time recruiting patients, finding patients, a single-arm trial might be beneficial here. So we'll talk in detail a little bit about some of these gains and losses. One of the main reasons that we would consider a single-arm trial is the sample size is typically going to be much, much smaller than what you would need if you were running a randomized trial, because you're only going to have one arm. So instead of having to randomize subjects between two arms, you just have one arm and you need about half as many subjects. It's also one of the big reasons is it's a lot easier to enroll patients in a single-arm trial, potentially. So if patients are unwilling to be accrued to a trial where they know they have a chance of getting a placebo arm, and maybe they would prefer to enroll to a trial where they know they're going to get an experimental therapy, it may be hard to enroll to a randomized trial because patients just don't want to commit to that. So those things make the single-arm trial beneficial, but some of the losses, and we'll go through in detail and describe these, one is that you need that historical estimate that you're going to compare to. So you need to have some assumption about what your control arm rate is. And as we'll see, that if we're not accurate about that, if we are off in that estimate, that we can see some severe losses in terms of our power and our type 1 error rate. And one of the other big issues with single-arm trials is we've violated one of those kind of pillars that Kurt described at the beginning of the session today. We've lost blinding. And because of that, we can see some biases, both from the patients, they know they're receiving the experimental treatment, and also from their assessors, their clinicians, who also know that these are the patients that received experimental therapy. So we lose that randomization, we lose the blinding, and because of that, we start to see biases. So one of the innovative trials that we are going to talk about now is how can we use historical information that we have, but do it in a way that we're not running a single-arm trial and all of the caveats that come along with that. And what we're going to do is borrow historical information in a statistical sense. Essentially what we're going to do here is augment a control arm in a randomized trial with historical data. And what that allows us to do is now, instead of doing just a one-to-one randomization, so half of our patients get control, half of them get the treatment, because we're going to now start borrowing information from the historical data, we can adjust that randomization ratio. So instead of a 1-to-1, we can maybe do a 2-to-1, a 3-to-1, or even more extreme, so that more of our patients are receiving the experimental therapy, fewer of them are receiving the control arm. But because we're using that historical data, that helps to augment our data on the control arm. So the benefits that we get from this is we retain blinding, so we now still have that, we remove some of those biases that we might see in a single-arm trial. The other thing is that since we are still collecting data in our trial about control subjects, that acts as a verification. So if we know what the historical data was and we get data in the trial, we can see how closely those two things align. So in a single-arm trial, we're just assuming that we know it. In a randomized trial, we get to compare and see how similar or how different was the control data in our current trial to the historical data. So historical borrowing, and we'll talk through a little bit of the details of how this happens. The gains here is that because we're using that historical information, we require fewer subjects in our randomized trial. So the sample size benefits compared to a standard 1-to-1 randomized trial are significantly smaller. So that lowers our risks. And we talked earlier about how single-arm trials may be easier to enroll if patients are averse to receiving a placebo arm. They may not want to enroll to a randomized trial. So here, if we're able to run a randomized trial that does borrow from the historical data, it can potentially be easier to accrue because we need fewer control subjects. Most of the subjects that enroll to the trial are going to be assigned to the treatment arm. So that helps to mitigate that risk of our accrual. being slower than expected. As before, if we were doing a single-arm trial, we would need a historical estimate. If we're going to borrow from historical data, well, we need the historical data. So we're relying on having some information, either from a previous trial, maybe a registry that we can use. And the other thing is, so what this design is kind of built on is how closely the current control data is to our historical data. And we're kind of banking on that being true, that these two things are similar. If it turns out that our current control data differs from what we've seen in the past, we have a limited backup plan for what to do if that doesn't match. So I'll talk through a concrete version here. Suppose that we're going to run a single-arm trial, and we're looking at a dichotomous outcome. So this is maybe a responder rate kind of an outcome. And what we would like to show is that our responder rate is greater than some estimate P0. So P0 is essentially our historical rate that we're going to compare to. So where do we get that? So one option is that we have an expert or a panel of experts who agree that 30% is the right number, just based on their expertise. It could be that we've seen some data from a small natural history study, so from patients who weren't treated or who were given kind of a standard treatment. So maybe in this small natural history study, we've seen 3 patients that responded out of 10 patients. So we have about a 30% rate here too. Other ways that we can get information about this control rate, maybe we look at retrospective chart reviews. Maybe we have some data from a large clinical trial. So for example, suppose there was a trial that had 200 patients and 60 of them were successes. Again, that comes out to about a 30% rate. But of course, if we were able to enroll a large clinical trial, there's no reason why we should have to run a small trial now. So now suppose that we're going to test the hypothesis that our response rate is bigger than 30%. And our rule here was we're going to run a trial with 20 subjects. And if we see 10 or more of our subjects that respond to the treatment, we're going to declare that trial a success. So if we apply this rule, it turns out that our type 1 error rate, so if our rate is truly a 30%, our type 1 error rate is about 5%, 4.8%. And if we assume that our treatment has a 60% rate, and we're comparing to that 30% on the control, so we have a 30% improvement, this rule of 10 out of 20 successes, that gives us a power of 87.2. All right, so we're done, right? We have over 80% power, we have 5% type 1 error rate. But the problem here is that we've made an assumption that 30% is the correct historical rate. And there's not always a lot of certainty around that. If we look at the literature, we'll see a lot of times in clinical trials that are run very similarly, there's variability in what that rate is. So in these characteristics that I just showed you, a 5% type 1 error rate and an 87% power, this is all predicated on that 30% being the right rate. So we've chosen that, then we've assumed that it was true. But what happens if that 30% is actually not the right rate? So we said on the previous slide that our type 1 error rate was about 5%, 4.8. So what we've done when we calculated that type 1 error rate is we assumed that our true response rate on the treatment and our true response rate on the control arm is 30%. And it turns out that if we run a trial and we count 10 out of 20 successes, then that gives us a type 1 error rate of 4.8%. But this definition never asks whether 30% is the right rate. So suppose instead that both the treatment and the control arm are both a 40% instead of a 30%. So here again, there's no difference between the control and the treatment. It's a null scenario. But now instead of both being at 30%, they're both at 40%. What happens? Do we think the type 1 error rate is still 5%? So it actually turns out that if the true rate is 40%, the probability of a successful trial under that rule that we've created is no longer 4.8%. It's actually gone up to 24.5%. So this is our type 1 error rate. if in fact our treatment and control are both 40% instead of 30% that we assumed. And it gets worse from there. So if we're even more wrong, so if the truth is actually a 50% rate on both control and treatment, our type 1 error rate goes up to almost 59%. So we're nearing 60. So the graph here, what this is showing is on the x-axis is the true control rate. So remember, we assumed that we had a rate of 30%. So that's this horizontal green line right here. And if in fact that is true, our type 1 error rate is 4.8%. So that's where this line crosses that green threshold. So the green line here is a 4.8%. So what happens is as our truth starts to differ from what we've assumed, as it starts to get bigger, what we see is the type 1 error rate starts to go up. And the further away from 30% that we get, the more we increase our type 1 error rate. What happens to power? So unfortunately, our troubles aren't limited just to type 1 error rate. We also have bad things happen to our power. So remember, what we were looking for was a 30% difference. So 30% on the control arm versus a 60% on the treatment arm. And we had seen that we had about 80% power if the truth is that our control rate is 30%. So what happens if our control rate is actually smaller? So what if the control rate is actually 20%? And we get an effect of 50%. So we still have a 30% difference from 20 to 50. But what happens is now our control rate has drifted. And what happens, as we'll see on the next slide, is that our power starts to go down. So now on the y-axis, instead of type 1 error rate, we're looking at power. Again, if we looked at our 30% truth on the control rate, what we were seeing is 87% power. But as our true control rate starts to go down, and we still assume a 30% improvement, our power is now less than what we had assumed. So we have bad things happen on both sides. We start to get increases in our type 1 error, we start to get decreases in our power, and this is all because we've made an assumption that 30% was the right rate. And when we're wrong about that, bad things happen. So why did we run a single arm trial in place of
Can we pause for questions?
Yeah, okay, sorry. Are there questions, comments? No. Okay. So this idea of our type 1 error rate and our power changing as a function of the control rate. Why didn't we talk about this when we run a randomized trial? Why am I talking about this only when we're talking about single-arm trials? Well, we can see what I've done now is added a line to our plot that shows what happens if we did a randomized trial. So now we still have a 20-patient trial, but instead of all 20 of those going to a treatment arm, I've now randomized 10 of those patients to treatment, 10 of them to control. So there's a line that may be difficult to see. It's an orange line right here that shows the type 1 error rate across the same range of control rates. And as you can see, that orange line is always below our 5% rate. So in a randomized trial, the type 1 error rate, in the tails, as we start to get really far away, we see that actually go down. But across the range of control rates, our type 1 error rate is controlled. So we've limited the chance of a false positive here. That red line, that was our single arm trial that we saw just a minute ago. What happens for power, again, we've added this orange line. This is now our randomized trial, is again fairly steady, fairly flat over the range of control rates. It does start to drop, but the other thing that we see here is the single arm trials. Over a wide range here, the single-arm trials has higher power, and that's really a function of the sample size. So the randomized trial, we have fewer subjects on our treatment arm. So the main point behind this is that history, in this case, we've assumed something about our control rate, leads us to the best guess of our parameter, but we could be wrong. Sometimes history leads us astray. what we see is that our best properties for our trial occurs when the truth is close to our best guess. So if we have this sweet spot where if our assumption is close to the truth, we get good behavior. But when our truth starts to differ from what we've assumed, we start to get biases. So if our truth is actually a lot different, we can either see inflated type 1 error rate, or we can see some big losses in power. All right, so now what we're going to transition to talking about is how we can use historical borrowing, so use an innovative statistical method to run a randomized trial so that we get those benefits of randomization, but try to control, not needing a large study. So now I'm going to consider an example. We're running a phase 2 trial, and our sample size, the most that we can afford, is about 210 subjects. I'm going to allocate my subjects in a 2-to-1 randomization. So out of those 210 subjects, 140 of them are going to go to the treatment arm, and 70 of them are going to go to a control arm. And then I'm going to use historical data to kind of augment that data that I get on the control arm here. So suppose that we have this historical data. So maybe this was a previous study that was run on the control therapy. And out of those 120 subjects in that previous study, 72% of them were responders. So that's a 60% rate from that historical study, and that's what we saw in the control arm. So I'm going to show you 2 examples here. I'm going to show you a trial that kind of ignores that historical data, and then I'm going to show you a trial that uses that historical data and just pulls it in with the data from our current trial. So we take those 72 responders that we got in our current trial, and we add them to whatever we see in our new trial. All right, so we're going back to this graph again. So the green line here, this is a 5% type 1 error rate. What we see, this is our trial that pulls the data together. So we take those 72 responders that we got on the previous trial, we add that data to what we get in the new trial, and what we again see is inflation of our type 1 error rate. Remember that historical data had a rate of 60%. So if the truth is 60% and we get data around that same number in our current trial, our type 1 error rate is actually a little bit lower than 5%. But as the truth starts to differ from what we saw in that historical data, we again start to see this inflation of our type 1 error. So what happens to the borrowing? So here, again, there's a reference line here at 60%. The purple line is what happens if we pool the data together. So we take those 72 patients from our previous trial, we add them in with our new patients. What we see is that power starts to increase when we do the borrowing as compared to a randomized trial that doesn't do the borrowing. So the orange line here is if we just do that 2-to-1 randomization, but we ignore those 72 patients that were responders on the previous trial. So if in fact our true control rate is 60%, so it's on point with what we saw in that previous study, we see these big gains between pooling that data together and ignoring it. If our true rate starts to differ from that 60%, though, we see the power for the pooling to start to go down quite substantially, where it starts to plateau if we ignore that data. So what we've done in this example was we've just decided to completely 100% use the data that we saw in the previous trial. We can also think about rules that allow us to use that data, but in some sense downweight it, so that we're not using the data to the full extent. So in what we call these are power priors. What these are static weights that basically say, okay, I'm going to take that historical data and I'm going to weight it by some amount. So a weight of 0 here would correspond to no borrowing. So I ignore that historical data. A weight of 1 corresponds to pooling, so that's what we saw in our previous example. But now we can allow weights that are in between. So for example, you could assign a weight of 50%. So what that means is each of those subjects in the historical data set essentially counts as half of a subject in the new trial. So you're kind of accounting for the fact that that historical data is potentially different in some way than in your new population. So this is an attempt to recognize the potential for drift in your population and account for the fact that subjects in the new trial might be a little bit different from subjects in the past trial, and so we're going to weight them a little bit differently. So for our statisticians in the room, the way that we handle this in Bayesian methods is what's called a power prior. So what we get is if we were pooling the data together, what happens is that historical data essentially just gets treated like it comes from the current trial. So we take our prior times our historical likelihood times our current likelihood. What happens when we do a weight that's in between 0 and 1 is essentially we're taking that historical data and raising it to a power. So what happens is if the weight is equal to 1, then this becomes equal to pooling. If weight is equal to 0, then this historical likelihood goes away. And anytime we have a weight that's different from 0 or 1, that essentially just adds some kind of a weight to the historical data. So how do we choose a weight for W? So common choices here might be half or a third. So again, this is an attempt to recognize that patients in the current trial are somehow different than patients in the past. So we're going to weight them maybe a half or maybe a third of the patients in our current trial. As we start to get values near 0, that essentially becomes like ignoring the historical data. It's possible that we could use values of bigger than 1. So what that does is essentially add more weight to that historical data. So if we have a really large weight, what that essentially does is act like a single arm trial. So it makes the current data that you've collected not as important. It gets overwhelmed by that historical data. So essentially, assuming weights bigger than 0, assuming large values of W is essentially equivalent to assuming that you have a massive data set from the past and you essentially know the answer. All right, so now what I've done on this graph is show different values of W, so different ways that we could weight the historical data. So let's see, so what we saw in the past is our purple line. This is right here. That was if we just assume that the historical data counts just like our current data. So a weight of 1 is that purple line. So this is our type 1 error graph, and as we saw before, we have this inflation as we start to differ from our 60% rate. But now what we see is that this inflation of our type 1 error rate is dependent on how much we weight the historical data. The more weight that we give to the historical data, the more type 1 error inflation we see as our rate starts to differ from the truth. But as we put less weight on the historical data, we see less of that type 1 error inflation. So this line right here, for example, is weighting the historical data at just 10%. And we see that we still do have some type 1 error inflation as our rate starts to drift away from 60%, but it's much lower than if we just treat our historical patients the same as our current data. So what happens to our power? So again, I'll point out the line we looked at before. It's a little hard to see, but here's our rate if the weight is equal to 1. So again, we see this kind of continuum of these lines that the less we weight our historical data, the more we start to approach the randomized trial. And the more we weight our historical data, the more we start to see these drops in our power. But again, most of the time, as long as we're close to our historical rate, as long as the truth is close to the historical rate, we see this spot where our power is always increased by the borrowing relative to doing a trial that ignores that historical data. So how do we choose a weight for W? How do we know what's the right way to weight the historical data? What we would really like to do is have some mechanism that if the true parameter is close to the historical data, we borrow a lot and we put a lot of weight on that historical data. But if our true parameter is different from the historical data, then we would like to downweight that data and not count it quite so much. The problem, of course, is that we don't know what the true parameter is, so how do we decide? Well, as in most cases, the current data is a good way for us to judge. So what we're going to do is let the data help us decide how much we should weight the historical data. So if we're running our trial, and in the current study, the data comes in around 85%, and remember our historical rate was 60%, well, that tells us that something is likely changed. The parameter is likely different from that historical rate, and maybe that's a situation where we don't want to weight our historical data quite so high. So any method that we can consider that assesses the agreement between the historical data and our current data is what we call dynamic borrowing. So what happens is we assess that difference between the current and historical data, and we decide on a weight that's based on that agreement. So as in most things, no method is foolproof. So it's possible that the current data leads us astray. So what could happen is that there's high drift. So really the historical data differs from the truth, but just randomly our current data happens to agree with the historical data, and we decide to borrow a lot, even when we shouldn't. So that's possible. It's also possible that there's really, in truth, no drift. The historical data is very much on point with the truth, but just for some random reason, the current data randomly deviates, and so we decide to not borrow so much when we should. Question, yeah.
Yes, if you don't mind me asking a question now. Mm-hmm. So when are you making the decisions about the weighting? Is this being done in an iterative manner or not? And then the second question is, what about a contemporaneous cohort? Would that be similar to historical bothering? Sometimes it is bothering. Because it sounds like quite a bit of work, actually, and I don't know about that.
All right, so the question is, when do we decide how much to borrow? So what we're going to do is actually create a statistical model that kind of does this automatically. So when we run the analysis, the statistical model does this assessment to see the agreement between the historical data and the historical data and the current data, and automatically adjusts the degree of weighting. Does that answer your question? You can do this borrowing just once during the trial. You can do interims where you assess it multiple times. But anytime you do the analysis, it would reassess that. Mm-hmm. Was there another question in the back? Yeah. No, I was actually pointing. Oh, okay, thank you. Okay.
The follow-on question with to that would be, though, you're designing the trial up front with an N and trying to figure out how many subjects you need, and at that point you don't take the historical borrowing into account, right, when you're preliminarily designing the trial, or do you? Because the question is, if it doesn't turn out as your interim analysis or your continuous analysis actually shows you, then what does that do to the initial trial design, and how do you sort of make up for that? I think that would be, the follow-on question to that.
Okay. Yeah, so I think if you're considering a design where you're going to potentially borrow from this historical information, that should be part of the pre-specification that you do. So you should be considering this up front when you design the trial and write down the analysis plan. And what else was I going to say? Totally forgot. Yeah, Kurt, you want to say something?
Hi. I think I got
Okay, yeah.
So another aspect of this is somewhat getting back to this insurance policy idea. What you're gaining here is essentially upfront, you're making an assumption that by using this historical data, I've seen multiple clinical trials, they all come at a 30% rate. I expect to see another 30% rate. I'm going to save 30% of my sample size by doing it this way. What you would want in your design is it to be prospective, that if I can go from 1,000 to 700, I use 700. But if in fact they don't agree, it does make sense to add this possible extension prospectively into your design, where at the beginning the protocol says, I'm going to go to 700, this is the analysis. If certain conditions aren't met, then the trial will automatically go to 1,000. You could also, we've designed trials where we've randomized 2 to 1, making use of the fact that we're borrowing these controls. And if in fact they don't agree, not only does the trial potentially get larger, but it also switches to one-to-one randomization because it's not borrowing those controls anymore as much. So all of this is, we're making an assumption, we're trying to make use of it, but you have this prospective backup plan, which makes the trial larger if the assumption's not met.
So I'm going to ask this at the risk of having been late. So I apologize if you've already gone through this. But this makes tremendous sense in terms of trial efficiency. What I worry about is if the natural history of the disease or the care is changing, then you are going to be slow to recognize that by continuously borrowing from prior data to inform your existing trial. Is there a way of handling that? Is there a philosophy around that concern?
Why don't you show the dynamic borrowing graphs? So.
Okay, so I think some of this will be addressed in what we're about to show. So the question is really about the drift, and if there is a drift, and potentially if it's slow to materialize, are you going to be able to recognize that during the course of the trial? I think that's part of the question. All right, so I'm going to go through some of the details of how we actually implement this borrowing. So the methodology here is called Bayesian hierarchical models. What these models do is they explicitly have a parameter that assesses the variability from study to study. So in our case, if we have our current study and we have the historical study, what the model is doing is assessing the variability in the response rate from the historical data to our current control. And this parameter that actually measures that variability has a direct relationship to how much weight we assign. So if the variability is small, so if our current data looks a lot like the historical data, we have small variability, the model's going to borrow a lot, and we're going to make heavy weight on that historical data. But if that parameter for the study variability, if it says, oh, these two studies differ very widely, then that means we're going to have a smaller weight on the historical data. So in the model, as we estimate this parameter, and we do that through Bayesian methods, we get a posterior distribution that determines the weight. So as we saw, the more borrowing happens when you agree, less borrowing happens when you disagree. We do have some statisticians in the group. The mechanism for this, so in general, we're going to let PC be the the control rate in our current trial. So C here stands for current. And then we have potentially more than one historical studies. So in the example, we just had one data set, but we could potentially have more. Maybe these are several small data sets that we've looked at that all have the same kind of control treatment. So P1 through P big H, these are the rates from our historical studies. What we do is we model the responders. So Y is the number of responders on our current data, and we assume that's binomial. Historical data also has a binomial distribution. And then what we do is we assume that this set of historical control rates has a distribution. Here we do the modeling on the Logit scale. And then what we do is we assume that this group of historical rates has a normal distribution with some mean and some standard deviation tau. And this parameter tau is what we just talked about. That's the parameter that measures the study-to-study variability. And that's what's going to control the degree of our borrowing. So tau, this is the most important parameter for the borrowing. If we just assumed that we knew tau in advance, and we don't estimate it from the data, but we just assume that we know it, that basically corresponds to giving a specific static weight. But in Bayesian methods, what we're going to do is actually estimate tau as we start to collect our data. And then what happens is as we start to estimate tau and we see that there's a high study-to-study variability, we start to downweight that historical data. But if we see that there's agreement between our two studies, then we start to increase our borrowing. So what that does is it creates what we call dynamic borrowing. So the degree of borrowing is not pre-specified in advance, rather the model is pre-specified in advance, And then as we accumulate the data, we estimate that variability, and the model determines how much we borrow, and it assesses that drift. So I think we've talked about that. So what are the properties of this model? So we previously looked at graphs where we had a static weight, either a weight of 1 where we fully pulled the data together, or a weight of 0 where we completely ignored it. Now what I'm showing, this bright lime green line, this is what happens if we do a hierarchical model. So we now allow the data to dictate how much of the borrowing we do. So on the left, this is our type 1 error graph. What we see is as the truth starts to differ from that historical data, we do start to see some type 1 error inflation. But importantly, that type 1 error inflation is bounded, and it eventually starts to go down. So what happens is the model starts to recognize, as you get way out here so far away from your true parameter, it starts to recognize, hey, these two things don't look the same. It starts to downweight that historical data, and your type 1 error rate starts to come down. And similarly, in the power, we also see mild power loss compared to downweighting. So here's the historical borrowing. And you can see that compared to if we just pool, we do lose a little bit of power. But if we compare to a design that ignores that historical data, that's the orange, we do see improvements in our power. In terms of power loss, so if our true parameter differs from the data that we're borrowing from, again, if we just pulled that data, what we see are these huge losses in power. If we ignore the data that kind of plateaus, the historical borrowing, it starts to plateau. So as your current data starts to differ too much from the historical data, the model recognizes that, starts to downweight the historical data more, so you don't see these dramatic drops in the power as we did before. So I'll quickly go through an example of this. So we had a trial that was, it's originally designed as a non-inferiority trial with a fixed sample size of 750 subjects, one-to-one randomization, so 375 subjects per arm. There was some historical data that was available. There were actually 2 historical studies here. If we use the historical data and we use the hierarchical model, what we found is that we could do a trial that had 600 subjects rather than 750, so about 20% fewer subjects. Here we changed the randomization ratio to 2-to-1. Instead of 1-to-1, we changed it to 2-to-1, so put 400 patients on treatment, 200 on control. And for the expected drift, so within the range where we thought there might be some difference from the historical rate, the type 1 error rate was controlled, and we actually had comparable power to our original design. So we were able to save 20% fewer subjects for about the same power and about the same type 1 error rate. Let me pause again for questions and see. I wanted to make sure I addressed your question. Questions?
May not be appropriate for this discussion. It's more of a philosophical one that if the natural history of the disease that is the focus of the trial is actually changing, then the assumption around how close this trial is to very good, very well-run multiple studies with event rates before, one shouldn't assume that this trial should be weighted less in its new control rate than the prior ones. And if we all do that with all of our trials, we're going to miss the idea that the biology is changing or that the treatment effect is not what it was 10 years ago.
I think what you raise, I think the key point here is, what is your expectation? There's a sweet spot where there's a benefit, and outside of that sweet spot, there's a loss. And really, it's an expectation of what is our belief on how often we're going to hit that sweet spot and how often we're going to have excessive drift on there. And in a lot of cases, we expect the sweet spot to be likely to be hit. And that doesn't mean every time, but enough of the time that this is worthwhile in repeated use. I think kind of the other philosophical point, if I think the natural history of the disease is changing quickly, I also have the worry, if yesterday's trial is not relevant to today, then today's trial may not be relevant to tomorrow. And so I have that other issue of going forward as well. So I think that becomes just a really hard problem to deal with. And this isn't designed to solve that. I think, oh, okay.
I think this is a more naive question than Neil's, but it comes down to the same issue, because I think the issue that maybe a lot of us would be struggling with is, when do you even consider doing this? Because it sure sounds like it could save a lot of time, and certainly from an NIH perspective, a lot of money, if someone was able to use historical borrowing. But when do people consider this? One, Neil raised the question of, the changing natural history of the disease. Another question I would have is, do you even consider it, for instance, if the historical data is retrospective, historical cohorts? Do you even consider, versus, let's say, a prospective cohort? Do you consider observational study data versus randomized, prior randomized clinical trial data? Because a lot of the examples that you've been using have been other randomized, controlled trials. In some of our, rare diseases where we want to use a lot of these innovative methods in design, the best historical data we have may be retrospective, cohort data. It might even be, if we're lucky, some observational prospective cohort data. It's less likely in some of our rare diseases to be another randomized trial. So, do you even think about using this in those kinds of situations?
So it depends. One of the key issues, so if you go to the FDA perspective on this, they're very upfront that they fully accept the statistical methodology. And the hang-up is essentially, do we trust the historical data that's being brought to bear, which is exactly the right question to ask. It does depend on the setting. There's a pharmaceutical group called Transcelerate, which they've been collecting. Essentially, a bunch of pharma companies have agreed to data dump a repository exactly for this purpose, among many others. But this is one of their key work streams. And they're writing some white papers on under what conditions do we think this is valuable. What that white paper will say is essentially you need to look at the areas like glaucoma, for example, very stable over time. PCR rates in breast cancer, very stable over time. Depression studies, you can get lots and lots of variability, and essentially it's so large this might not be a good idea, so you really have to know the area. In terms of rare diseases, the smaller the trial you're running today, the broader that sweet spot. There's so much variability in the study that anything you can do to reduce the variability is bigger than any risk you have to creating a bias. And so in rare diseases, you have the interesting trade-off that the data might not be as nice, but because the sweet spot is so large, it can be in your benefit. So one thing we want to do is when we design this, we write out the sweet spot is this big. You need to be comfortable that your rate is within these bounds or this isn't going to work for you. And then it becomes, I do not want to call it a gamble, but in the long run, it's do you believe the sweet spot? And if you're going to be right 80% of the time, then this is worth doing. If you're going to be right 40% of the time, then this is not. And it's a matter of obtaining that confidence and repeated use.
And I think I didn't ask the second part of my question very clearly now that we've gone through some of this, but to get away from the problem of the historical patients as we've been discussing, could you use this methodology for a contemporary control that's not included in the study but is an observational cohort or on another registry? So that's what I was trying to get to.
If you have something to say, go.
I'll say it. Yeah, I don't want to.
I was thinking. No, go.
So this is a dear to my heart area. So contemporary, the general issue of drift. One part of that is separation in time. Other parts of it are going to be different sites, different standard of care, other aspects. So I think that the general notion of how close they are, time is one aspect of that, and other factors also come into play. So contemporaneous gets rid of the time aspect, but may not get rid of different sites, other differences between what's going on. We've had a lot of use. We run platform trials, which are 10-year continuous trials. Those are nice because you can see the whole time course, and you can get an idea how much have they been changing over time. And you've got almost this self mechanism for checking this assumption. So contemporaneous, I think they're good, but it doesn't eliminate all the possible discrepancies. Let's see, I think we need to take a break. Dore is informing me that we've got goodies in the back. We're going to take 10 minutes. Will that work? Let everybody get some food. If it takes 12 minutes, we'll take 12 minutes. So let everybody get some food and rest.
We're going to go ahead and get started back up, so feel free to grab your coffee and your goodies. All right, so we're going to get started back up again. So this morning, the previous session, we mostly talked about ways that we can efficiently use our resources. So we've talked about ways that we can make our sample size flexible, ways that we can use data that we have in hand to help make our trials more efficient. One of the other broad topics that we want to talk about is how innovative trials can help us to answer broader questions. So a standard randomized clinical trial requires focus. So typically what we see is a trial that is a one-to-one randomization looking at one treatment versus control. We have a very specific homogeneous patient population that we're looking at. We've made an assumption about our treatment effect. And a lot of times our standard randomized trial doesn't look at a lot of different things, like we don't look at treatment combinations, we don't look at different what we call domains. So there's reasons historically for why we have this focus in our clinical trials, but one of the things that we often think about when we're in the process of designing a trial is we try to anticipate if the trial doesn't come out the way we hoped, if we have our p-value at 0.06, for example, what would we regret and what would we think we would have done differently in our trial? You know, would I think, oh, I should have looked at a different population, or I should have had a bigger sample size, or, you know, I should have looked at a different dose. And so we try to think about what are those things in our trial that we might regret if we don't do them. And if we can think about those things in advance, and then we can make those changes to our design up front to help reduce that anticipated regret. So innovative trials, rather than focusing on a small question, often aim at answering a bigger question or a set of questions. So because of this, we can broaden our focus so that we're not just maybe looking at a single population or maybe not just looking at a single dose. And this makes our trials more robust. So robust meaning that we're robust to some of these uncertainties. So if we have a narrow focus and we're just designing a trial with one dose versus our control arm, you know, we're making an assumption that we've picked the right dose. Or if we just look at a population, you know, of certain characteristics, we're making an assumption that that's the right population where the treatment benefit occurs. And if there's uncertainty about that, if there's uncertainty about what the right dose is or what the right population is, and we choose incorrectly, there are you know, obvious consequences to that. So if we can make our trial more robust and looking at broader questions, multiple doses, multiple populations, not only does that help with accounting for the uncertainty that we have and maybe giving us some protection against making a wrong assumption, but it also allows us to look for things like interactions between treatments that you don't get to do if you just selected one single treatment. So this idea of looking at more questions, this is not a new idea. So a lot of us may have been told that, you know, when you're running a clinical trial, you can only change one thing at a time, right? So, but actually, you know, R.A. Fisher, one of the giants in the statistical world, recognized in 1926 even that, you know, asking a very narrow set of questions is not necessarily the best idea, and if we can answer logical or ask logical, well thought out questions, that we have a better chance of answering those questions. So one of the designs that we can talk about to ask broader questions is a factorial design. So the idea here is that instead of asking to compare just one single treatment versus control, we may be interested in combinations or different types of treatments that in combination with one another. So for example, we may have a trial where we have maybe what I call a domain. So domain A, you can either give big A or little a. So maybe this is, in the example we're going to use, maybe this is patients who are on a ventilator. So you have patients that's either on a ventilator, big A, or not on a ventilator, that's little a. And maybe you're also interested in another treatment, antibiotics. Patients are either on antibiotics or they're not antibiotics. So that's treatment domain B. Patients can either get big B, which means they're assigned to antibiotics, or little b, which means they're not. So, you know, a trial that just looks at ventilator versus no ventilator, or a trial that just looks at antibiotics versus no antibiotics, that would be a trial with a single narrow focus. And what that doesn't allow us to do is look at any interactions between these things. So what we could do instead is look at a trial that looks at the combination of these things. So you could do big A in combination with big B, big A in combination with little b, a big B and a little b. So in the example that we used, we just had two domains. You can generalize this. So maybe our example is not just a ventilator and antibiotics, but maybe it's a ventilator, antibiotics, and steroids. We now have 3 domains, and each of those domains has 2 possible treatments, with it or without it. And we could run a trial where we look at all of those different combinations. So now we've got ventilator, yes, antibiotics, yes, steroids, yes, versus ventilator, yes, antibiotics, no, steroids, yes. And looking at all of these things together allows us to answer a broader question. So this design is called a factorial design, and these types of designs are always going to be better than a separate trial for each variable because it allows us to look at the interactions between those two things. So for example, if you were running a trial where you were just looking at steroid use in combination with antibiotics, so big A, big B, and versus neither of those things. So a patient either gets both or they get neither. That doesn't allow you to answer the question of whether the antibiotics are important or whether the steroids are important. So if you see a benefit in both of them together, which one is it that caused the benefit, or is it really only when they're together? And you can't answer that question unless you do a factorial design that allows you to look at that interaction. And let's see, so if the factors really do interact, only a trial that looks at all of them, only a factorial design is the answer to finding that interaction. If these factors don't interact, you can still estimate the effects of all of the factors, and the nice thing about a factorial design is that the cost is actually similar than if you just explored all of them independently. So an example of this type of design is called the PROSPECT trial. This is a pediatric respiratory trial. It's an NHLBI funded trial. This was looking at the comparative effectiveness on two factors or 2 domains. So the first one was the positioning for a ventilation. So patients were either supine or prone while receiving the ventilation. And then the type of ventilation was either conventional or a high-frequency ventilation. So now we have, in combination, there are 4 possibilities. So we have 4 arms in our study. And the trial was designed to have to have interims where we could consider either dropping a row or dropping a column of our table. So potentially we could have a situation where we stopped all of the arms that were in the prone position and continued the supine position, or we could do the opposite, where we continued the prone and stopped the other. We could also drop one of the rows. So for example, you could stop all of the arms that had the high frequency ventilation and continue the conventional, or do the opposite and stop the conventional and continue the other. So this is an example of a factorial design that allowed us to look at these different domains. So as we said, the factorial design is really the only way that you can detect these interactions if they exist, the only way that we can account for which of those factors contributes to our treatment benefit. These trials can be slightly harder to plan and implement when you have multiple factors. But the nice thing is that if you don't have interactions, it's almost free to do this, so why not? And if the interactions are there and you want to estimate them, this does typically lead to a larger sample size. So that's one way that we can address broader questions, is a factorial design where we look at multiple treatment domains. Another way we can look at broader questions is by doing a dose-ranging study. So many of the trials that we see today have small number of treatment arms, maybe 1 or 2 doses versus control. So we have often made an assumption that those are the right 2 doses that we should be looking at. But if we make a guess, it's a possibility that we're wrong, and maybe the best dose isn't actually one of those 1 or 2 that we've chosen. So a dose-ranging trial, what this is going to do is a trial that incorporates more doses. So 3 doses, 6 doses. We've looked at trials that have many more doses than that. A lot of times when we do a dose-ranging trial where we have multiple doses, we want to have a model that explicitly looks at the relationship between the efficacy and the dose level. And a lot of times we'll often look at adaptive features, such as maybe eliminating arms that aren't performing well, so we can start to drop arms, or maybe change the allocation to our doses. So if some doses start to look like they're performing better, we increase the randomization to those arms. So we can have adaptive features, and we're going to talk about those a little later this afternoon. We'll talk about something called response adaptive randomization, or RAR, and give an example of that. So that was a little teaser. So we're going to talk more about dose-ranging trials, but wanted to talk about response adaptive randomization before we go into that. So the gains of this is, well, first of all, we have a better chance of picking the right dose. If we can look at a broader set of doses instead of just picking 1 or 2, we have a better chance that we've picked the right dose in our trial. So the losses, a lot of times we think if we're going to add more doses, that's more arms. Isn't that more patients? So if I'm going to add another dose, does that mean I now have to, almost double my sample size? And the answer is usually actually no. So with modern modeling, so when we add these dose response models, when we start to add features like adaptive randomization, there may still be a small increase to the sample size, but it's actually a marginal cost. So the cost of going, from 3 arms to 4 arms is actually not that large. So one of the other things that happens with dose-ranging trials, we often pick a dose-response model. Again, that's an assumption, basically looking at what's the form of the relationship between efficacy and dose. So that does rely on an assumption, so that has to be carefully considered. So we'll circle back around to dose-response modeling in just a moment. But first wanted to talk a little bit about enrichment. It's kind of a buzzword lately. So the idea is sometimes we don't know the right population of patients to enroll in our trial. An enrichment trial recognizes that there's some uncertainty about the population and which population actually has the benefit. So an enrichment trial, you can do this maybe one of 2 ways. You can either start with the largest population and then start to narrow that down and start to focus on the population where you're seeing benefit. Or you can go in the opposite direction. So maybe start with a small population, and then as the trial moves on, you start to expand into a larger, broader population to see if that benefit still holds. Some examples of this. We've seen this in a lot of oncology trials, in particular when we're looking at a targeted therapy that's tested in multiple tumor types. So the idea is that maybe it's not as important where the tumor originated, but rather the molecular target of the agent and whether it's going to be beneficial in patients with those types of tumors. So in oncology, we see these trials, we sometimes call them basket trials, where instead of just running a trial in this histology and then running a separate trial in lung cancer and running one in breast cancer, what we can often do instead is enroll a broad population. So we enroll patients in different types of tumors, and then we use our methodology to try and figure out which of those populations is there a benefit. We've also seen an example, or some examples in other areas, stroke, and we're going to walk through an example here where the enrichment was done to determine the time window between when the stroke occurs and when the patient gets to the hospital, and the stroke severity. And the idea is, does the effectiveness of the treatment differ across this population, or is it effective everywhere? So enrichment trials, the main idea here and the gain is that we can identify where the treatment is effective and where it's not. One of the downsides is that we do have some more complicated statistical methodology to account for the fact that we've got multiple groups, and if we have multiple chances to win. So we have to account for that. But one of the big gains is that this is robust. So because we're looking at a broader population, there's a better chance that we're going to ultimately end up with the right population instead of just guessing at the outset and potentially getting it wrong. These trials tend to be larger than if we were just doing a trial in one homogeneous, focused population. But then, of course, the potential benefit of that is it might be easier to enroll because we've now got a broader inclusion-exclusion criteria. So an example of this is a trial called Dawn, run by Stryker. Jeff Saver was the PI of this trial. And this is investigating the use of a device called Trevo for the treatment of stroke. Thanks to Roger Lewis, who provided these slides for us. So before the trial was started, there were some papers that came out showing that endovascular therapy was highly effective in some, but the population for where that benefit happens was a little unclear. So which patients are most likely to benefit from this treatment? So at the start of the trial, the investigators, they sat down and tried to think about where are there uncertainties about this device? What are the things that we would like to learn in the trial? So this slide shows kind of the set of uncertainties that they listed, things that they don't know that they would like to learn. One of those was, where is the benefit? Where does this occur? So in stroke, the primary endpoint here was the modified Rankin scale, or MRS, disability scale. So this is a 7-point scale, kind of an ordinal scale. So 0 means a patient has no symptoms at all. A 6 on this scale means a patient has died. So the question was, is the treatment most beneficial for subjects who are maybe in the lower range of that scale, or more beneficial for patients who are in the upper range, or is it beneficial across the entire range? So that was one of the things that we didn't know going into the trial. Another question was about who benefits. And one of the things they wanted to look at was the core infarct size. So this was, the way I understand it, was a measure of the damage to the brain tissue. And one of the hypotheses was that only those patients with smaller core infarct sizes would benefit from the treatment. But those with the larger infarct sizes maybe were less likely to benefit. But that was an uncertainty we didn't know. And that was one of the things that we would like to learn about during the trial. One of the other uncertainties was the magnitude of the benefit for this device called Trevo. Is this, does it have no benefit? Does it have a large benefit? Does it have a small benefit? And the implications here were, well, if it has a large benefit, we could run a small trial. But if the benefit is smaller, we would need a larger trial. So this is kind of the idea that we might need a flexible sample size because we have some uncertainties about the size of the treatment effect. So for all of these questions, if we were to just make an assumption and say, okay, we're only going to enroll patients who have the small core infarct sizes, that would be making an assumption. And if that assumption is incorrect, that could have consequences for the trial, that maybe the device is beneficial and we just picked the wrong population. So what we have done instead is designed a trial that's going to look at all of those factors. First of all, we're going to talk about that first one, the MRS scale. So this is the 7-point scale. Typically in the analysis of these types of trials, the scale is dichotomized. So for example, we look at patients who had an outcome of 0, 1, or 2. So these are good outcomes. Patients have small to no symptoms. And we count the rate of patients who have an outcome of 0, 1, or 2 versus those that have 3 through 6, and kind of a responder rate analysis. So the downside of that is that if the benefit occurs, if we move patients who might have otherwise been a 4 and we move them to a 3, this kind of analysis that just lumps together the 3s and 4s doesn't account for that. So we miss kind of the subtlety and the treatment effect there. So one way to handle that is to have an analysis method that looks at that scale. So it's not just looking, does a patient go from somewhere 3 to 6 down to somewhere 0 to 2, but looking at how they move and what degree of movement they have. So what we've done here is constructed what we call a utility-weighted MRS. So what this does is it takes each value of that MRS score, 0 through 6, and assigns it a utility. So here, utilities that are, a utility of 0 is essentially there's no benefit, and a larger utility score means that there's a bigger benefit. There were a couple of papers in the literature that had proposed utility-weighted scores. So these top 2 rows in the table shows some utility weights that had been proposed. So for example, these were on a scale from 0 to 10. So a utility of 0 is no benefit to the patient. A utility of 10 is a good benefit. So patients who end up with a MRS score of 0 were assigned a utility weight of 10, so that's a good outcome. Patients who died were assigned a utility of 0. 5 is kind of an interesting one. So 5 on the MRS scale is essentially a patient who is in a vegetative state. So one of these proposed methods treats that, gives it a very small utility score of 0.1. The other proposed method actually gave this a negative utility score, so that this outcome was actually considered worse than the outcome of 6, which was death. And then you can see that in these the other range of the MRS, the 2 proposals, they agreed pretty closely. So in the DAWN trial, the new trial, they actually used the weights that are shown here. So outcomes of 5 and 6 both received a utility score of 0, MRS of 0, got the good utility of 10, and then the various degrees in between. So then when the analysis is run, that allows us to look at a little more subtlety of where the treatment effect is. So instead of just doing this dichotomous, looking at 3 to 6 versus 0 to 2, we can now have a better idea of the magnitude of the benefit. So for example, a patient going from a 4 to a 3, that's a pretty large increase in their utility, whereas going from a 1 to a 0 has a smaller change in their utility. All right, so that was one question that we addressed. The other question was who benefits, and this idea of should we enroll only patients who have small infarct sizes, or should we enroll a broader population? So the infarct size, this was thought to be something they could identify as an eligibility criterion for entering the study, and might define different populations and how they respond. So the strategy could be that we start with the small infarct sizes and then expand if we see benefit there and start to enroll the larger ones. The other strategy, what we call enrichment, is we start by enrolling the broad population, and then we start to restrict the enrollment criteria if necessary. And the DAWN strategy actually did this later one. So they started by enrolling up to 50 cc core infarct volume, and then they allowed enrichment rules. So they had interim analyses that would allow you to then prune off certain sections of that. So they could potentially decrease that from 50 to 45, from 45 to 40, et cetera. And the way that was done was by looking at the likelihood of a positive trial. And if you were more likely to get a positive trial from decreasing from 50 to 45, then that was done, and you enriched to a smaller population. And I think I have another slide about that in a moment. So the third question that we had some uncertainty about was the magnitude of the benefit. So ideally, if the truth is that this device doesn't work at all, if there's no benefit, we would like to be able to stop the trial early and save that sample size. Or we would like to say, okay, if there are certain regions of the population where there's no benefit, we would like to stop enrolling just in that population. But if we see benefit in other areas of the population, we would like to keep going. So if there's no benefit, we want to be able to stop. If there's a small benefit, but it's clinically important, we would like the trial to keep going. So they set a large maximum sample size of up to 500 patients, but then had interim analyses that allowed us to stop early. The other idea behind a clinically important difference and understanding what that means was related to the choice of the outcome. And instead of just using the dichotomous measure, trying to pick an outcome that's going to be more sensitive and recognizing where that benefit is. And finally, if we have a large treatment benefit, that means we don't really need the full insurance policy, as it was described earlier, of a large trial. So we'd like to build in rules that allow our trial to stop early, and potentially as early as 200 patients if it's likely that the trial is going to be successful. So everything about this design was pre-specified. They actually wrote up a methods paper describing their methods. So the sample size was a maximum of 500, and they had multiple interim analyses, starting at 150, 200, all the way up to 450. And they pre-specified what types of decisions could be made at each interim. So some interims, they had the possibility of enriching. Some interims, they had the possibility of stopping the trial. The rules for early stopping were based on the predictive probability of success. And then the rules for stopping for futility were based on if the probability of success in any of those subpopulations was less than 10%, or sorry, in all of those populations was less than 10%. The enrichment criteria here was based again on the predictive probability of success. And the idea was if we could increase the probability of winning the trial by restricting the population and if by doing that increase the predictive probability by at least 10%, then we would enrich. And we would eliminate populations based on their core infarct size if they had less than a 40% chance of benefit in that population. And the enrichment here was done from the top, so we started with infarct sizes from 0 to 50. And then we would calculate what's the predictive probability of success with the full population. What's the predictive probability of success if we restrict it to 45 and below? What's the predictive probability if we restrict it to 40 and below? And if by doing that, enrichment could increase the probability of a successful trial, then we would enrich. So some of the statistical details around this. So this, as you can see, became kind of a complicated trial, the enrichment, the early stopping. So we had to do a lot of simulations and make sure that the trial was going to behave the way we wanted it to. One of the aspects that we looked at was what is the criteria for a successful trial. So we were calculating the posterior probability of the MRS utility-weighted MRS score being better than the control arm. If no enrichment happens, so if all through the trial we enroll up to 50 cc core infarct volume, then the probability of benefit has to be at least 98.6% to be positive. And this threshold takes into account the multiplicities of our early interims for early stopping. And that's if no enrichment occurs. If enrichment occurs, we actually need a more restrictive criteria because of the fact that we've kind of cherry-picked which population we're going to be analyzing in the final analysis. So we started out with 98.6, but if enrichment occurs, there was a rule for how we would adjust that threshold to account for the enrichment. We had some statistical models, kind of similar to the models that we've talked about before, that shared information across infarct size populations. So they had a model looking at the benefit as a function of the infarct size. And we ran, before the trial started, massive computer simulations to show that this all worked, that we controlled type 1 error, that we could understand how the enrichment happens. So here's the paper. This was published in the Journal of Stroke for the DAWN trial. And this is a trial it has actually run. It actually finished and reported out in January of this year in the New England Journal of Medicine, and actually saw quite significant efficacy across the wide range of patients. So I believe the outcome was it never actually enriched, but the trial did stop early and was successful at an interim, and I don't recall which interim that was. It was the first one. Okay, so they saw wildly positive results at their first interim and were able to stop early. Pause to see if there are questions on the enrichment. No. All right, I'm going to hand it over to Kurt to talk about platform trials.
Okay. This one's mine. Ooh, I need the clicker. Thank you. All right. I'm a pacer, so I've had instructions. There's actual tape on the floor. I'm not meant to go past the tape, so I'm going to sit here very carefully. All right, so I want to talk about platform trials. These are trials that enroll multiple treatments within the same structure. So we're investigating lots of things at once. They're motivated by the notion that if you look at a disease, and I've got Alzheimer's up here, there's been lots of Alzheimer's trials. Unfortunately, they've all failed. What's happened is if you take, I have an example here, 25, there's been more than 25. But as a society, what we've done is we've invested 50 to 100,000 patients in Alzheimer's. We've failed over and over and over and over and over again. And we've invested 1,000 to 2,000 patients in each novel treatment, and we've invested 50,000 patients in 25 to 50,000 patients under control. The net result of that is we know a ton about the natural course of Alzheimer's. And do we need 25 to 50,000 patients enrolled on control in Alzheimer's, or would it have been valuable to do a few less and maybe test a few more drugs while we were at it? We could have had more shots on goal, so to speak. So what platform trials are aimed at is trying to make that happen. So this is just a graphic. This is really how we've allocated patients. A whole bunch of controls, and I mean, certainly there's a lot of patients in each of these bars, but the relative proportions, this is a little off from what we would think would be the natural thing to do. So a platform trial is a special example of something called a master protocol. Master protocols are kind of another buzzword these days. Essentially, they're a protocol that, imagine a protocol with no drug names. Sounds kind of odd, but if you're investigating Alzheimer's, there are certain endpoints that you're interested in. Patients are visited at particular times. They have certain medical tests, medical procedures. You can write a lot of the protocol without really knowing what the experimental drug is. We're going to be testing ADAS-Cog, and we're going to do it at this rate, and we're going to do this kind of analysis at the end. So the master protocol is really specifying all of that stuff, the visit schedule, the endpoints, the analysis, and so forth. A master protocol is designed so that it can be run over multiple treatments, and in particular, it can be run perpetually. So you can say, I'm going to start a trial in Alzheimer's. There actually is something called the EPAD trial in Alzheimer's, which is a platform. And at any given time, there are 3 to 6 treatments in EPAD. It's still getting going at this point, but there will be 3 to 6 treatments in EPAD. And drugs can go in and out of that trial. And when a drug comes in, what happens, instead of having to write an entire new protocol, you say, look, most of this stuff, we're going to have the same visit schedule as all the other drugs, the same endpoints, the same tests. But when a new drug comes in, instead of writing an entirely new protocol, what we're going to do is we're going to add an appendix. And that appendix says, this is what we're going to do for drug X within this. So usually that appendix gives details of what the treatment is. So certainly all the medical information, any specific safety risks, any particular safety concerns, if you have to do extra kinds of testing. We have to do a certain kind of blood test for this drug that we wouldn't ordinarily have to do. and if there's any patient exclusions. We ran an Ebola trial, for example, and some of the drugs could be used on pregnant women and some of them couldn't. So the appendix would say, this one's not applicable to pregnant women. A platform trial is a specific form of master protocol, and it's defined by its being continuous. So certainly most master protocols are intended this way, but some of them we're going to have 6 drugs from the start. A platform trial is usually intended to run for a long time, and drugs are going to enter and exit over time. It's also intended to have a cohesive inferential structure. And so what do I mean by that? I'm not trying to run, platform trials are often confused with cooperative networks, where if you run a cooperative network, you've got a large patient infrastructure, but every trial that comes in is really separate. So I'm going to run this trial and I'm going to get patients from this network, and I'm going to run that trial and I'm going to get patients from the network, and so on. But the trials really aren't talking to each other. These are trials where they aren't trials stapled together. What happens is patients, when you come into this trial, if there are 6 treatments running in it, you can get randomized to any of the 6 treatments. So it all has a single umbrella that decides randomization, and it has a single umbrella that is that describes the analysis structure. One of the key reasons for doing that is if you have multiple arms and you've randomized across everything, that allows you to pool controls and it allows you to compare across different arms because you had randomization, as opposed to if you didn't, if you just ran this trial in this part of the network and that trial in that part of the network, you'd worry about potential biases because you didn't have this randomization. Yes?
So, Kurt, that's very interesting. Are there, in these master protocols, as you're describing them, do the inclusion criteria for each of, let's say, the sub-trials that are going on within the master protocol have to be the same in order for it to work? Because I could envision where you might be able to have a master protocol for a trial, but the inclusion criteria for trial A might be different than B, C, or D, just maybe even slightly.
So there are a couple different directions you can go, and the efficiency that you gain depends on the details. So I'm trying to remember, what's the European oncology trial? If you ask me any other time, I'd think of it immediately. All right, anyway, there is a trial that for some reason I'm blanking on. What it is designed to do is there are several different oncology treatments, but every one of them is intended for a different molecular mutation. So they've got drug A is intended for mutation A, drug B for mutation B, and so on. That is really more of they are using the platform as a screening mechanism. If you imagine these trials run separately, trial A would exclude out 80% of the people they screen. Trial B would do the same thing. Trial C, because they're just looking, each one's looking for a small segment with their particular mutation. If you do all of these together, then what happens is that screening mechanism has gained you operational efficiency. Other trials are intended to look at the same treatment and you've got multiple drugs that work on the same population. That gives you inferential efficiency because you can share controls among that. So it really, you can go both directions there, and the efficiency you gain, you get efficiency, but it's a very different kind depending on which direction you go. Oh, yes, sir.
How do you get the companies to agree to a master protocol, which, I don't know, maybe you can point to a reference or something that you can point for the lawyers to kind of debate about. And so the second question is more realistic, is there a type of randomization platform or schema that you would recommend for a platform trial where it's more efficient to bring in new drugs as other drugs kind of get exited?
How do you get companies to agree? Very, very carefully. So The first thing that you can always do is you can always get a company to agree to be the second, the third, or the fourth drug into the platform, because you can effectively promise them the cost is cut in half. The thing that you can't ever do is convince someone to be the first drug in the platform trial, because they're difficult to get together. Now that's a little bit tongue-in-cheek, but that part's absolutely true. The other sticking point, which I think you probably were intending, is companies often don't want to play in the same sandbox, period. One thing that I think is fascinating about platform trials, there's a direct comparison between drugs within a platform, and you're randomizing between them. Usually what has happened, platform trials nowadays actually have part of their charter is a statement that they will never publish and compare the drugs together. And often that is enough. I-SPY 2, you asked for papers. I-SPY 2 has been running for about 10 years. It's had lots of companies go through. I don't know if there's any publication that describes the interactions, probably by design, as we never actually want to describe the private interactions. There is a paper that's been submitted from the Adaptive Platform Trial Coalition, which is self-named, but Derek Angus is leading that, Derek Angus and Brian Alexander. And they, I think, have some discussions on how to get companies to buy in. So I'm not giving you a good answer because it's a delicate question, but we have successfully done it for a number of trials. An example of this, so let me just give you this is the efficiency. So we've got an emerging epidemic. This is inspired by our work on the Ebola epidemic in West Africa. Multiple possible treatments were suggested for Ebola. If you remember, there was something called ZMapp, a number of other antivirals. There was some suggestions certain cancer drugs might be effective, and people were thinking of combinations, all kinds of stuff going on. So one question was, if you have this large space of possible treatments, how do you look at those? What do you need to do? What's the right way to evaluate 20 different drugs? We've done this in Alzheimer's as a society over the past 20 years. We've done drug A failed, drug B failed, drug C failed, drug D failed. We've run a lot of trials in sequence. So how do we look through these 25 different issues? In order to make the problem a little bit more concrete, suppose that under the standard of care, there's a 30% survival rate. That's consistent with our Ebola example. It might have been closer to 40, but I will say 30 here. We're going to make the assumption this is a hard disease, so I'm going to assume that only 10% of novel treatments work. 90% don't. And that depends on what area you're in. If you're in Alzheimer's, nothing's worked in Alzheimer's, probably assuming 2% might be optimistic. Sepsis, an incredibly hard area. Certain kinds of cancer immunotherapies right now, unfortunately, are in much better situations. You might expect a larger number in certain areas. But I'm setting up this problem, so 30% chance of survival, and of this large bucket of possible treatments, only 10% work. How do I find the golden nugget among those large numbers? That 10% of novel agents that work, they have 50% survival. So working here means I increase from 30 to 50. All the rest do nothing, so 30-30. So certainly things could be harmful, I'm not assuming that. So how do we search? So this is what we might do in a traditional trial. We'll pick one, we'll run a trial. I'm going to do 100 patients per arm, so I do 200 patients, 0.025 type 1 error. If that first trial is successful, I go, yay, I found a treatment, and I declare the process a success. If that trial fails, then I'm going to pick another drug out of this bucket, and I'm going to run another trial. So this is just intended in a sequence. Grab a drug, run a trial. If it works, great. Otherwise, grab another drug. If that works, great. Otherwise, grab another drug, one after the other after the other. Now, this is actually a pretty good strategy if you can pick what the absolute most promising drug is in advance. Generally speaking, that's hard to do. If you actually knew which drugs work, you probably wouldn't need to do the trials. So this, if our bucket has 10% good drugs, what happens? So on average, we've got to run through 9.8 treatments to find a successful drug. That's about in line with our 10% of the drugs work. Our N, so this N is the total number of patients we had to treat, 1,966. So, and that's effectively, really we're running 10 different trials on average in order to find that one golden nugget. Let's see, we assumed an accrual rate, so we found one in 12 months on here. So here's another way to do this search. Instead of looking at one-to-one over and over again, well, what does that do? My first one-to-one trial, I enrolled 100 controls. My second one-to-one trial, I enrolled another 100 controls. The third one, I ran another 100 controls. That's the same control over and over again. So that doesn't make a lot of sense. Let me at least combine those. So I'm going to do a shared control design. I'm going to do 5 agents at a time, so still 100 per arm, but now 5 agents are going to get compared to a single control arm. And again, I'm going to do pairwise independent analyses. So now I run 600 patients, 100 controls, 100 on each of 5 active arms. If I find a successful drug, I go, yay, I won. Otherwise, I go back and I enroll another 5 arms at a time. So I run this sequence. This is good. And I get savings here from sharing the control resource. The fact that I haven't enrolled 100 controls over and over and over again is what's gaining me efficiency. So my average sample size, it now takes me 1,528 patients in order to finally find a successful treatment. And I can do this in 8 months rather than 12 months. I can do a little bit better. Remember we talked about futility earlier, being able to stop bad drugs? I'm going to do that. So those 5 active arms, if some of them aren't performing well, I'm going to stop them for futility. That gets me an additional benefit, a large one. So that 1,528 I can drop to 971. So now I'm almost in half. So sharing controls using futility, I can now find an effective treatment twice as fast. What a platform does is it adds another piece to this. So we're still going to investigate 5 agents at a time, but now what we're going to do is we're going to run interims every 150 total subjects. I was about to get up, but I refrained. So the futility for an arm here, so we're still declaring futility. What you can think of this platform as doing is whenever an arm stops for futility, I'm going to bring a new arm in immediately. So I'm not running a 6-arm trial and waiting for it to finish and then running another 6 arms. Whenever a slot opens, I'm going to bring a new drug in absolutely immediately. So I'm just investing things forward. Question?
So is there a point in this mechanism in which your controls can cross over? If it's somebody having repeated events?
Can someone, can somebody, so that depends. Some of these are designed to allow crossovers. There's a platform trial called PanCan, which it allows patients to be enrolled on multiple therapies over time. In other ones, in this one, we actually did not allow crossovers. Usual issues with crossovers on washout effects and carryover effects would apply to this, but in theory, it's possible.
So like if you had asthma, for example, and you wanted to see what was the best abortive agent in the middle of an asthma attack, and someone was on the trial as a control, there was other agents, after some period of time, the controls could switch after they'd been controls for X amount of time, because the events are in themselves isolated, although the patients are the same.
Okay. I don't know that much about asthma. But it sounds plausible. That sounds like the pan-can. As long as you don't think, if I've been on drug A or control and I switch to drug A, if you think that's no different than being on drug A, that usual question, if the answer is, if you could do a crossover in a regular trial, you could do a crossover in a platform.
So I just wanted to touch on actually go back to the eligibility question for a second here, because, if you're replacing one of these arms, let's say the new treatment comes in with, for example, you can't give it to pregnant women who have Ebola. How does that modify the overall exclusion criteria for kind of the platform trial? Does it have to change? Are they not eligible to be randomized to one of the arms? Kind of how's that handled operationally?
Yeah, oh, there's a follow-up. I'm a little nervous now. So no, you're good. So generally speaking, it's complex. What you would often do is have to analyze, say, pregnant women separately. In the Ebola trial, the two special populations were pregnant women and children. And it was actually an interesting debate because the pregnant women issue, the obvious question, if you die of Ebola, that's bad for the baby. So it was a huge debate over which drugs actually ought to be excluded for pregnant women. You'd have to make a modeling decision. Do you want to combine the pregnant women and the normal data, not normal, it's the wrong word, pregnant women and everyone else together in the analysis and pool it when you're evaluating an arm? Do you want to view pregnant women separately when you do it? You'd have to basically come up with a statistical model to account for that. And I don't have an easy answer for you in that situation.
From an enrollment perspective, though, as they come in, they wouldn't be eligible to be randomized that arm, though.
Yeah, so the operational issue is easier. If you had arms A, B, C, D, E, and you said that C and E were ineligible for pregnant women, then a pregnant woman coming in would be randomized to A, B, or D. And so potentially you've got biases in those populations, and that's where the modeling would come into play.
And so then with kind of changing inclusion criteria potentially for treatments and also different treatment modalities, I mean, is these essentially are unblinded trials then?
Depends. So the EPAD trial in Alzheimer's, for example, some of the treatments are pills, and some of the treatments are injections. For the purposes of EPAD, you can get a placebo pill or you can get a placebo injection. So the way EPAD works is you are randomized to arm A, B, C, D, or E. If arm B is an injection, then you're randomized. You have a chance of getting a placebo injection or an active B. If you're randomized to C and it's an oral, you might get a placebo oral or you might get active C. So it's kind of a two-tier randomization. It's a question of whether, in EPAD, they actually pool the oral placebos with the injection placebos, which was actually a controversial choice. What was interesting, one of the compelling arguments that was made is we've spent 20 years and haven't been able to move the needle on Alzheimer's. Somehow, if we could give an injection as opposed to a pill, and that will affect Alzheimer's, that would be a little strange. So, but it is a choice on whether you think that different placebo modality matters, and it will make life more complicated. So I haven't given you a direct answer. We try to deal with it as best we can. All right, let's see. Okay, so this open platform. So again, you can think of the adaptive plus every 50. This was the essentially the 5 at a time, but then we waited for the trial to finish. This is when we invest the resources forward. So now you can see we're down to 849 subjects. So we're searching for this needle in a haystack. We've gone from 1966 down to 849. We've saved about 60% of the subjects, and we've really done it in multiple ways. We've shared our control arms, done multiple treatments at once. We've employed adaptive features, and we've invested any savings that we've gotten forward as best we can by bringing in new drugs. This is in a paper, this reference in Clinical Trials. It has essentially this table. There are lots of examples of this. They have different features. There's a paper by Janet Woodcock and Liz Levange in New England Journal of Medicine, really touting these. These are the examples in the paper. So some of these, I-SPY 2 has been running for about, I think it's 8 years, but it's been running a long time. LungMap's another oncology trial. ADAPT is really a concept trial for resistant antibiotics. This is not the Ebola trial I was describing. We did something for the Gates Foundation. This is something with the NIH. This is another Alzheimer's trial separate from EPAD. And then this is an antipsychotic. There's actually a whole bunch of, so these are some more examples. I don't intend to go through these slides. The only thing this slide is meant to indicate is, this is getting back to your question, there are just lots of weird complexities that happen. And really, some of these are blinded and some are not. Some of these are embedded in a learning healthcare system and some are not. And all of them involve quirks to deal with the issues. And there are occasionally trade-offs. We will pool the placebo arms. We don't think it's going to make a big deal difference, but we can investigate twice as many drugs. So we've made that trade-off consciously. Certainly, these are a challenge to set up. So if you're going to do a platform trial, getting the first drug in, it is a long haul convincing companies to play in the same sandbox, to convince researchers to play in the same sandbox. There are operational aspects that are more complex. Informed consent is often two-tiered. Often there's an informed consent. Are you willing to be randomized into a drug? And then separately, are you willing to be randomized to the placebo arm or the active arm of this drug, is how EPAD does informed consent. So you have to do it twice, for example. On the plus side, the second and future agents, this is a no-brainer thing to do. If you had these platform trials set up, it would be ridiculous to do, if the whole world were set up where here's a breast cancer platform, here's an Alzheimer's platform, here's a heart disease platform, here's a stroke platform, it would be ridiculous for anybody not to enroll in these platforms and go run their own trial with their own controls. What you've got here is a warm base, sites are up and running, there are larger networks allowing you to find subsets of patients. Everything is better in this particular world. And I think that is good. All right, so just as a conclusion, again, these things, it's the startup cost versus the massive savings at the end is really what the trade-off is here. All right, Anna. This one's still mine. Okay.
All right. So the next topic is about changing the randomization ratio during the course of a trial. So the idea behind this is, instead of doing a 1-to-1 randomization, or if we have multiple arms, 1-to-1-to-1 randomization, the idea is that we'd like to efficiently focus our resources to the arms that are most promising. So the place where we'll typically see these is dose-ranging trials, for example. That's the example we'll talk about in a moment. Platform trials, as Kurt just described. Factorial design, so if you can identify among multiple arms which arms are the most promising, and then start to focus your resources on those arms. Different ways this can be achieved. Kind of the simplest method in many ways might be considered arm dropping. So if you have sufficient evidence that an arm is not performing well, you get rid of it. Another way that this could be done is a little more subtle than arm dropping, but is adaptive randomization. So if you have an arm that's not performing well, you start to decrease the allocation to that arm and start to increase the allocation to the arms that are performing well. So you can see that in the figure. going to describe this by walking through an example. This is a trial that was designed for smoking cessation. The final endpoint is looking to see if a patient is still smoking in weeks 3 through 6 of the treatment. So the idea is in weeks 1 or 2, they may not be fully weaned off of the cigarettes yet, but by weeks 3, 4, 5, and 6, if they don't smoke in any of those weeks, they're considered a success, a failure otherwise. The other idea in this trial was that patients who drop out of the trial and maybe don't complete the full 6 weeks of follow-up are also considered failures. So this could be, we assume if they drop out, maybe that's because they are smoking again. So we consider those patients a failure. This trial has multiple doses. So in addition to the placebo, we have 6 dose arms ranging from 1 milligrams up to 100 milligrams. And what we would like to do, the goal of our trial, is to identify the dose that has the maximal response. So the dose that has the highest response rate. One of the quirks of this is that we expect that a U-shaped curve is possible. So the idea is that maybe those top doses may have higher dropout rates, for example. Maybe those higher doses come with more adverse events and patients are more likely to drop out at those doses. And because those doses, if you drop out, you're counted as a failure, it's possible that the dose response curve is not just increasing, but that actually at some point it starts to decrease because those adverse events start to outweigh the benefits. So in this particular case, we're assuming that U-shaped curve is a possibility, and we're looking for the maximum effect, which may be at one of the middle doses. Other trials, instead of looking for the maximum effect, they might be interested in looking for something like an EDX, like an ED90, which would be the dose that gives you 90% of the maximal effect. Again, the idea here is that as you start to get to the higher doses, maybe those are doses that have adverse effects. So if you can back away from that and give a slightly smaller dose and sacrifice only a minimal amount of efficacy, maybe that's the optimal dose to give. So one way that we often handle that is by looking for this ED90. So trying to find a dose that has at least 90% of the benefit, but maybe is not the highest dose. Another way we sometimes handle that idea is that trade-off between efficacy and safety is explicitly by using a utility function. And the idea behind this is to combine several endpoints. So maybe we can explicitly define that trade-off between the safety and the efficacy, and we do that with a utility function, and then we try to find the dose that has the maximum utility, which combines those endpoints. So our current trial is a little bit simpler than that because we're just going to count patients who drop out as failures, and so that aspect of that trade-off is just explicitly handled in the primary endpoint. Some details about the trial here. So we're going to enroll 280 subjects into our trial. In addition to our goal of identifying the best dose, we also would like to calculate, this is a phase 2 trial, we're interested in what's the probability that a dose that we carry forward is going to be successful in a future trial. So what we're going to do for each dose in the trial is we're going to calculate, suppose that we choose this dose and we carry this dose forward into a phase 3, 1-to-1 randomized trial with placebo, and in that future trial, we plan to enroll 400 patients, 200 on each arm. And what I would like to know is the probability that that dose is going to be successful. And that could be, used as a futility rule. It could be used as part of our success criterion. So I'll describe several kind of iterations of the design that we went through, starting with what we call a basic trial. So in our basic trial, we're going to enroll 40 subjects to each of the 7 doses. So equal randomization. This may not be the optimal strategy. Oftentimes when you have multiple doses, it's actually beneficial to enroll more subjects to the control arm. But here we're going to consider a simple example where it's just equal allocation to all of the doses. In our basic trial, we're not going to do any modeling of the efficacy across doses. Instead, we're just going to analyze each dose independently without any borrowing between the doses. So in order to do that, what we're looking at here is the response rate, we call PD for dose D, and we're just going to give independent prior distributions to those doses. So we're doing this in a Bayesian fashion, and what that means is at the end of the trial, our drug is going to be considered successful if our posterior probability of beating the control is higher than 99.2%. So right now, we don't have any adaptations, we don't have any early stopping. This is just a fixed sample size trial, 40 per arm, analyzing the doses independently. And the reason that we have this high threshold is a multiplicity adjustment. The fact that we have multiple arms in our trial, we have multiple shots on goal, so that we need to have an adjustment that it's harder to win each dose. All right, so we've described some scenarios that we're interested in. We'd like to see how well this type of design behaves under these different scenarios. So in the table here, I've got the doses along the top and a few different scenarios along the rows. So first of all, we'd like to see how well the design performs in the null scenario. We think that the control arm has about a 20% rate. So about 20% of patients that get a placebo stop smoking. And then the null scenario assumes that there's no benefit on any of the doses. It's 20% across the board. We've got a scenario we call harm. Here is where the lower doses are equivalent to control, and then as you start to increase the dose, the response rate actually goes down. We've got a set of scenarios where we have a treatment benefit. In all of these scenarios, the benefit increases with dose, so that the highest doses are more beneficial than the lower doses to various degrees. And then finally, we have a set of scenarios we'd like to investigate where the maximum benefit actually occurs somewhere in the middle of the dose range. So here in the inverted U scenario, the best dose is the 25 milligrams, and then the effect starts to decrease as you get higher doses. And then we looked at a scenario where that peak occurs earlier in the curve. So it occurs at the 5 milligram dose. So we'd like to see across this range of scenarios what happens to this trial. So in this table, what we're showing is essentially the power and the type 1 error rate, which is in this column here. So under the null scenario, our type 1 error rate is about 5%. If we look at the scenarios where we have a treatment benefit, this particular trial has pretty modest power here. So ranging from 14% up to about 56%. And in our inverted U scenarios, somewhere around 45 to 50%. So this is an underpowered trial. We don't have the highest power to detect these kinds of scenarios that we're interested in. The far column on the right shows the probability of picking the best dose, and that best dose is described here in parentheses. So for example, in the positive scenario, we had about a 40% probability of choosing the best dose. And in these inverted U scenario, we had about a 48% probability. So some of that depends on how many doses are good and what's the magnitude of the difference between your best dose and maybe the second best dose. All right.
And then that gives me 10 minutes for
Okay.
Jamie.
All right. I went the wrong way. All right, so now we're going to take that basic trial that we described, and we're going to add some modeling, and we're going to consider a dose-response curve. And what this is just a function that describes the efficacy as a function of the dose. Here in our example, we can't consider a curve that assumes a monotonic relationship, so we expect there's a possibility that the curve increases for a while and then plateaus or maybe even comes down. So we can't assume something like a logistic curve or an Emax curve because those have a built-in assumption that the effect increases with dose. So we're going to use a model called a second-order normal dynamic linear model, or NDLM. So this is kind of a smoothing spline function. So now I'm going to go back to that table where we're looking at the probability of success. So now in each one of these cells, we have two numbers. So the number on the left is our basic trial that we just looked at before. The number on the right is now we've done nothing else except add a dose-response model. So under the null scenario, we have about the same type 1 error rate, about 5%. But in some of our other scenarios, we start to see some pretty big increases to our power. So take a look, for example, at the positive scenario. Previously, we had 56% power. Just by adding on a dose response curve, we've increased that to 70% power. So we can see some pretty big gains. And the idea here is that we're able to borrow information across these doses. Let's see, inverted U scenario, we saw some jumps from 50% to about 64% by adding the dose response model. The probability of picking the right dose, we see some modest gains here, about 40% to 46%. Here it didn't change very much, about 55%. Another way we can think about, choosing the right dose or have we made the right decision is how many doses continue into the next trial. So what we're showing in this table is the probability that we pick the right dose given that we can carry one dose forward, the probability that we get the right dose if we carry 2 doses forward, and the probability that we get the right one if we carry 3 doses forward. And here, this is conditional on trials that were success. So if we just take the subset of trials that were successful, looked at which doses we carried forward, the trend here is obviously if we take more doses forward, we have a better chance of choosing the right one in that set that we take forward. So the next thing we're going to add to our trial, so we've looked at just going from a basic trial to adding a dose response model. Now we're going to start to change the adaptation. So now instead of just allocating patients equally across all the doses, we're going to do something different. So we're going to start the trial with what we call a burn-in period, where we have a fixed allocation. This could be 1-to-1. This could be some other ratio, but we're going to have a small portion of our trial that has this allocation that's fixed. And from that data, that initial set of data, we're going to estimate the dose response curve. And then for the next set of patients, we're going to change the allocation ratio and start to allocate more patients to the doses that have the best efficacy. And we'll continue to iterate this. So we'll have interims that are frequently spaced, and each time we do an interim, we're going to update those randomization probabilities. So how do we decide how to change these allocation ratios? So one simple method is for each dose, we're going to calculate what's the probability that that is the best dose, and we'll allocate in proportion to that. So doses that have the highest probability of being the best dose, those get the highest randomization probability, and doses that have lower probability of being the best dose are going to have a smaller weight there. So in our example, so to make this concrete, I should go back. So here, what we do is we calculate this probability. What you can do is actually also raise this to a power, which helps you control how aggressive you are. So if you calculate this probability and then raise it to a power of 2 or a power of 3, that allows you to be more aggressive about going after the best dose. In our example, we're not going to do that. We're not going to put an exponent on it. So what we're going to do is start with a burn-in of 10 patients per dose. And after that burn-in period, we'll estimate our model, and we'll update our allocation probabilities every 4 weeks. The other kind of bells and whistles, there's lots of different ways you can do this, is if any of our doses has less than a 5% probability of being the best, we're going to temporarily just zero out that dose so that it's going to receive no patients until the next interim. And then we'll reassess that probability at the next interim. So effectively what that does is a temporary arm dropping, but it's not a permanent arm drop. So it's just zeroing out that dose, and then as we start to get more data, we'll reevaluate it. In these trials where we do adaptive allocation, how we treat the control arm turns out to be really important. So remember here, our final analysis is analyzing relative to control. So we're comparing the response rate of our best dose to control, and in order to make a good comparison there, we need control patients. So we want to make sure that we don't allocate away from control. For example, we don't want to just put the control arm in with the adaptive randomization and say, well, if the control arm starts to perform poorly, we allocate away from it. If we start to allocate away from control, what that does is it hinders our ability to make comparisons to the control at the end of the trial, and that hurts our power. So here what we're going to do is fix the allocation to the control arm. So 2 out of every 8 subjects are going to be allocated to the control arm, and then the remainder of those subjects in the block will be allocated adaptively. So here are our results. Now we've got the basic trial, the NDLM trial, and now we've added the adaptive allocation. And we can start to see some large benefits again, especially look at here, the low is the best dose. So now by allocating adaptively, we've gone to about 50% power, 47-ish, to over 60% power. We see some gains in our other scenarios. The positive scenario went from 70 to 79% power. So because we're now making more efficient use of our resources, putting the subjects on the doses that we care about, the ones that are performing the best, we see some gains in the power. So again, some of the gains here ethically, the idea behind adaptive randomization is to avoid exposing patients to arms that aren't performing well, and instead give the patients who are enrolled in the trial the best chance of getting allocated to a good arm. So these trials can be a little more complicated to set up. Careful planning, careful calibration, making sure you're not making decisions too early, and some operational details about doing these interims and changing the adaptive allocation ratio. So those have to be considered as well. Questions?
All right, last topic. Everybody tired? Okay. So I want to talk about rare diseases and more informative endpoints. So I'm going to do this by way of an example. This is a G&E myopathy, which many of you may know more about than I do. But essentially, this is a progressive disease where your muscles are gradually replaced by fat. And it goes essentially from your feet up. So what happens early in the course of disease, you basically lose muscles around your ankles. It moves up to your knees, through your hips, through your arms, and it is eventually fatal. Very rare, worldwide prevalence, 4 to 21 out of a million. There's nothing that helps this particular treatment. This is challenging to do, or nothing helps particular indication. This is a challenging disease to do research on. And the reason is, while the prevalence is low, in addition, people are at different stages of the disease. So people early in the disease, an endpoint like 6-minute walk might be relevant because they're losing function in their ankles and they're having trouble walking and so forth. People late in the disease are not ambulatory at all. So 6-minute walk is irrelevant to people like them. You may be interested in the ability of somebody to grip strength or something like that late in the disease. So it's difficult to come up with a clinical trial design that accounts for all of that. So what we did is we came up with a disease progression model. So we essentially came up with a statistical model that described the entire disease course. We had natural history data on 38 patients. Each one of them is at a different place in the disease. So for example, this person, their strength is a lot lower. Each of these curves, I'm not going to go through all the details of this, but different kinds of their knee extensions on here, their elbow flex strength, their shoulder strength, knee flex. These are different pieces, components of the disease. You can see this patient has completely lost the knee and dorsiflex, but still has strength in other areas. So each of these patients, what we can do, I'm going to skip this model. What we essentially did is we took that disease course and we said, this is what happens to dorsiflex over time. This is what happens to your knees over time, to your grip over time, to your hip extension over time. And what we were able to do is we could map each patient onto their disease age. So some patients might be in this area where if we were going to follow them, this is in years. So if they're in this area, you can see the dorsiflex is the thing that's going to give us the most information. In this area of the disease, this is a situation where the hip extension or the grip gives us more information about the progression of their disease. So what we tried to do is take each person's data and tried to map it onto each of these. So this is the kind of fits that we got to the natural history data. So this is one subject analyzed over several years, and you can see essentially these are the observations, and this model is fitting it quite well. So this is saying if we know your 5 or 6 strength measures, we can essentially map this to you're at disease age 10 or 20 or 30, depending on where you are in the disease. This also includes the uncertainty bars around each of the different ones. You can see knee is an interesting one. The knee behaves very differently than the other measures. This is more fits. I recognize we're a little short on time. As an advertisement, Melanie Quintana's giving a webinar on disease progression models sometime early in the spring. There will be more detail on this in that webinar as well. But I'm still trying to indicate this is a model that fits very well when all said and done to the natural history data. And it fits well even though the patients are at different components of their disease. What we did in this is the trial design, those 6 endpoints. What we did is the endpoint for the primary analysis is the disease age. So for each patient, what we did is we took those 6 measures, we mapped it back to a disease age for each patient, and the primary analysis is basically measuring, can we halt the progression of the disease? If you come in at baseline and if Anna's at disease progression age 10, 2 years later, has she gone to 12 or is she still at 10? And if it were somebody else, if Doré came in at disease age 30, 2 years later, is she still at 30 or is she at 32? And so it was the time involved in that we were measuring as the primary endpoint. The trial has 50 patients. They're enrolled 3-to-1, treatment to placebo. And again, if you think of this as the disease progression, this is the overall disease age. What we're really trying to measure is, are we halting the progression of the disease? So it might be that Anna going from 10 to 12 over 2 years is full progression. Anna going from 10 to 11 over 2 years, we've cut the progression in half. That's our measure, our primary endpoint. The purpose of this is to combine all this information in the 6 endpoints on strength and get us an adequately powered trial, which this does. So this ends up, you can see if we've got a treatment effect of 50%, we've slowed the disease progression in half. The trial power is 98%. In fact, if you slowed the disease down to 75%, so it's 25% slower, we still have about 75% power. So this is, again, a trial that tried to work on a very small number of endpoints, and I recognize I haven't given it full description in order to, since we are running out of time here, but there is more on this. This trial is what came out of the NIH on rare and neglected diseases. It is currently being run by Neuronext, or about to be run by Neuronext as one of their trials. Let's see, so let me end here, make sure I have questions. Please sign up for the ICTR website. When you download slides, you can sign up for an account. Our email addresses are on here. If you have any questions, we'd be happy to answer those. And certainly enclosed, and maybe have any questions, comments to end. Thank you all. I think we're good.